Yeah, we done 20 years. We all started somewhere. Yeah. The guy said, yeah, and then we, oh, we talk about how I don't okay. So I didn't know what she said.
Did we get that last presentation? Yes. Sweet. And it's right here, the Murray WQ. Beautiful. Okay, and who is first? Oh yeah, that's right. Sorry. Multiple rooms, everything kind of gets jumbled together. All right, cool. There you go. No problem. Check this microphone.
But how sometimes some programs results which is basically the question not screening well, and like yeah, let me yeah,
I'll cut the Q&A off. I'll stop it. Like if people, I'll tell them if they have more questions, they find me afterwards to ask, which that a lot of people will probably will.
But and I will say my students that I've done a lot of emergency and we didn't have yeah, yeah, so there was a very
My
I was like, I thought it was the windows. I don't know if you use windows. I was like, support with the helping pros. I'm like, wait, we're gonna have a mouse. And then I'm like, wait. But like, also like, oh my god, how do I shoot the mouse? Because I'm in You're tiny. Okay, that is an ordinance. Yeah, you know what? I'll like tomorrow. Yeah. So.
Yeah.
So are you?
We're playing off of the so you're trying to have time, okay.
But one thing, there's only 4 segmentations in our in section.
Yeah.
So is that, so do you, is it that each get 20 minutes or are you keeping 15 minutes at the end for questions? And that's this is a terrible part because like
Yeah.
you
Yeah.
so do you want to, because I feel like It's not balanced here, but you want, it's not usually. You want yeah. And I'll do an hour 15, right? So steps we five. Oh, well, we can get 28, we can go in. Well, 17 is. Yeah. Because we don't do anything with the part we want to. Yeah. Oh,
I have not seen that in Korea, I've seen the thing, and honestly, they can still make it.
Yeah, for the, okay,
Yeah.
17.
Yeah, so yeah, they can-
Thinking about 12 minutes to the market, so like, I'm counting like 12 minutes.
Okay.
So we have 9 minutes.
12 minutes for the presentation, 5 minutes for the. We're just working out, you have 75 minutes and 4%, we don't have 5 minutes. So we're gonna get 17 minutes at least, 12 minutes for the presentation, 5 for 5. So that actually seems really, yeah. I'm very good.
So you have both credit, but once you.
I can just get it.
I just think that's good.
Does that work for you? Yeah, none of us knew.
And I'm like, oh, I'm supposed to be.
Oh, yeah.
No, well, no, it's
Oh, we gotta stop.
because some of these sessions are 5 and some are 3 and 10 for. I'm on the first.
So we were just trying. And then if there's lots of Q&A, I'm just going to say.
Oh, yeah.
How is this like the? This is like the Tufts.
We've done projects together.
I made of course a slide deck. So I want to make sure it's very. Barbara's, Barbara.
You just have to, it's not not generally. So, and I'm a lot of specific person. We're gonna have
All right. Good afternoon, everyone. Thank you for joining the verbal presentations. This room is presentations on microbial risk and treatment. Sorry, my name is Ryan Kronk. I'm with the Water Institute. It's great to have you all here for the second day of presentations. It's a great lineup. Tufts and friends, all four presenters. So, and up first we have Daniele Lantagne. Take it away.
Thank you. So today I'm presenting on a project, Household and Community Risk from Informal Industrialized Guano Farming Facilities in Cambodia. This project is part of a larger project called Stop Spillover, which is a USAID funded project that's been awarded to Tufts University and partners with the goal to work in countries to understand and stop the spillover of emerging infectious diseases. There's three components of Stop Spillover, and the project I'll talk about today is in the second component, which is on developing, testing, and implementing interventions and policies to reduce the risk of priority viral zoonotic spillover pathogens. So the Cambodia project, Stop Spillover, is a project in 7 countries. Cambodia is one of them. The Cambodia project, the priority viral pathogens are highly pathogenic avian influenza and zoonotic coronaviruses. And so the places that the spillover of zoonotic coronaviruses has been identified as problematic are density and distribution of host bats, in bat guano production facilities, and in farming practices. And here in the map of Cambodia, you can see Phnom Penh, the capital, and just kind of northeast of that, up the river, is Kampong Chong. So what is household bat guano production? So in Kampong Chong, there are households that install artificial bat roosts to encourage bats to roost, and then they collect the bat guano that the bats produce, and they sell this for fertilizer, and it's an income-generating activity, right? And so our goal was to understand artificial bat roost risks to communities and develop interventions to mitigate those risks from those bat roosts. So I have to say, when they first asked me to help on this project, I was like, how big could a bat roost be? I was like, maybe 6 feet by 6 feet. So here's a tiny one. It's a bat roost, and you see there's plastic on the bottom where they're collecting the guano. Okay, here's a small to medium-sized one, and you can see the water storage containers, the household, the laundry, the pipes, and the bat roost right there in the background. And then here's the larger scale size, the fully industrial bat roost. So we're talking about thousands to tens of thousands of bats roosting in these areas for the guano. So we did 2 concurrent methods. I'm presenting work that's a big partnership. And so there was a whole team that collected from the bat guano itself from the bats. So they did a survey on roost construction, operation and maintenance of the roosts, the size and production, the economics of these roosts. And then they also did the guano sampling. They installed infrared cameras to count the bats flying in and out. They did species identification with acoustic recorders. They collected bat urine and feces, and they tested that at a local laboratory for bat DNA presence and coronaviruses. Concurrently, we worked on looking at food, water, and surfaces. So we worked with a survey of both bat-producing households and their neighbors, which is something that comes out really important that we'll talk about through this. We did a survey on demographics, we did a survey on water, sanitation, and hygiene practices in these households, environmental health, and then bat exposures that these households had. And we also collected samples. We swabbed food and surfaces. We collected water samples, and we also took our samples to a local lab, and we also were planning to identify bat DNA and coronaviruses. So the results from the bat guano farm side of this, this sampling was conducted in April to May 2023. They surveyed 16 bat guano producing households. There was 256 guano and urine samples, and those coronavirus PCR results are pending right now. They resampled 245 households in August, and there's 2 more trips to resample, right? Now let's get to the wash practices. So one of the things we saw in these households is the wash practices were, there were a lot of potential risks for this guano to get into kind of transmission routes. So only 10% of people covered their drinking water, 45% covered their vegetable garden water, 40% self-reported treating drinking water at home, 20% covered their food or covered their meat while they were drying. Only 2% had a dedicated space for hand washing, and 20% used running water for hand washing. We actually surveyed 14 households that produced the bat guano and 14 neighbors. We had a fairly small sample size, so these are non-statistical results, but one thing to note is that the neighbors were less likely to have wood walls, a higher quality construction house, have a water tap in the home, to cover their water, to have a dedicated space for hand washing, to report treating their water, or to eat dairy. So I think one of the assumptions that could easily be made is these bat guano production facilities are done by people who are living in poverty to get some extra income. But in fact, the reality is these are highly industrialized and concrete structures that are done by wealthier people to collect even additional income, and are actually potentially putting their neighbors of lower socioeconomic status at risk without the associated economic benefits. So what do we have in our results? We have our laboratory results from our 408 food, water, and surfaces and 501 bat urine and feces. For coronaviruses, the lab in Cambodia was able to do that, and but not bat DNA, which we were not able to get, which is something I can talk about. But interestingly, our results are with the Cambodian government, and I am not allowed to report them. To the point where they're like, not only can you not put them in your presentation, but don't do what everyone does at the conference and then talk about it with all your colleagues after the presentation. The only people who are allowed to report the sequencing results from our testing from all this are Cambodian government officials. So this becomes an interesting thing when you start talking about academia and conferences, right? And of course, I will respect that, so don't ask me. However, what I can say is we have overall results. I'm going to read these out because everyone who knows me knows I don't like reading things on PowerPoint, but this is a USAID funded project and I'm required to do a few things by the project leaders, so I'm going to read some things out. Households invest a large sum to construct large artificial bat roosts that return a large value to the household. They make about $30 per bag of guano, and they're making many bags, many bags a night with these large roosts, right? Household members have good knowledge of bats. They know what bat types, they know how they're flying, they know the risks. And women are primarily responsible for collecting the guano, and they're often using just bare hands. They're collecting it without PPE. Bat guano is visibly present on food and surfaces. So when you go and you swab, you'll see bat guano on the food and surfaces in both farming and non-farming households, those neighbor households. And neighbor households had lower socioeconomic status than farming households. So when we talk about the whole goal of this was to identify potential risks of spillover and transmission, and so we want to get to our mitigation measures. Well, our recommended mitigation measures for our bat guano farms are using PPE during the harvesting of the guano, washing your hands, storing the guano safely, and excluding livestock and pets. These are households that often have cows, chickens around them from the guano production facilities. Now, the recommended mitigation measures for food, water, and surfaces: disinfect your high-touch household surfaces, wash hands frequently, cover foods that are left out to dry, either that you're drying meat or foods that you're leftover foods, and cover food stored in open kitchens, right? And one of the things is, this is not targeting that should just be done at the households producing the bat guano. It's targeting that needs to be done at the community, because the bat guano production puts the entire community at risk. So let's talk about some challenges and limitations of this. It's a small sample size. We did 10 households of each because we were swabbing a lot. We were trying to look at where in the household, we do know where in the households the contamination is, and we really wanted to look at a lot of samples per household. There's regulations which I very much support about kind of decolonialization of WASH, which is not going into Cambodia, taking samples out and bringing them to your lab back in the US. It does mean that some of the things that we wanted to test, like bat DNA, could be done at Tufts, but can't be done in Cambodia. And that's a complication of something. This essentially means we need more training of lab staff in Cambodia to do some of these methods. We're not allowed to disclose the lab results, as I talked about, because they belong to the Cambodian government and they're the primary, they can disclose those. Now, I also want to point something out, which is we did all this work, we wrote the report, we gave it to USAID, we gave it to all the partners involved, and this list is really boring. Wash your hands, cover your food, cover the bat guano, cover, none of this is sexy. This is incredibly basic, boring wash. 2% of people in these communities had hand washing stations. One of our recommendations is install hand washing stations. I told that to the partners and they're like, what will USAID say when we say we need to install a hand washing station? That's incredibly boring, right? So, in my summary, again, I don't like to read, but I am on this project. Informal industrialized bat guano farming in Cambodian households is a highly lucrative business that leads to spillover risks to farmers, farming households, and the surrounding community. These risks are disproportionately borne. Community and household hygiene interventions are needed to reduce those spillover risks, and WASH has a role in this, but it actually feels like WASH, it's pretty basic WASH, right? So USAID is the funder of this, so thank you to our donor. I have the contents are the responsibility of spillover and its partners and don't necessarily reflect USAID. And then our consortium partners, including, and I want to call out Tetra Tech, the local Tetra Tech employees. The person I worked with at Tetra Tech on this project who finalized the report and what I supervised actually has the exciting news that he got a postdoc, which means is why I'm presenting instead of him. So he's working on a postdoc. And that's my time. And I'm happy to take any questions about this if anybody has any questions. Yeah, Mark.
Yeah, I actually have a comment, so. Mark Zapsa, University of North Carolina and Aquagenics LLC. So in Cambodia, there are readily available small portable commercial hand washing stations, which you may know about because we actually got those developed. They're made in Vietnam, if I'm not mistaken.
Yep.
But they are widely available. So
A Happy Tap.
These people, Happy Taps. So these people should have no excuse in being able to get those Happy Taps and take them to wherever they work.
Yes, and I totally told the project about Happy Taps and gave them all the information. There's some concern that was raised about whether the Happy Taps were appropriate in this area of Cambodia. That's always an appropriate thing to raise, but also the concern is like this is a big viral spillover project and the Happy Tap is a pretty basic wash thing. And it's interesting to kind of talk about that with the, but I agree, like the Happy Tap is a fantastic project and people developed that.
And the other thing is that some people might object to the color of the Happy Tap.
Yeah.
But I think now it comes in different colors. So
Yeah,
if you
it
want
does.
a blue one, you can get a blue one.
It was green initially.
Green one, you can get a green one.
Yeah, exactly. No, thank you. And that's a great point because this can build on other wash projects that we've done. Exactly. Other questions? Yeah. Or you can be loud and I can repeat it. If you'd rather do that, I can repeat the question. Yeah.
How did you get them to become involved and what was going to be the feedback, especially with this extra barrier of
Right.
submitting?
Yeah, so so the question is how are the households involved and participate and how do we provide the households feedback? So the first thing is this is a 5-year project. These households have been approached via the community leader first and then the households themselves. And there's a kind of whole process of being involved in other studies. In this study, a consent process And so there's a long community building piece of the project that came before we went into a house in all our protective gear and swabbed their entire household, right? So there's kind of years of work building up to that. And that comes from Tetra Tech, who did that work, right? It's a local partner. It's not something I did. But it's a local partner. We did train government officials from Cambodia. There was a set of 4 government officials who came with Tetra Tech and myself so that they could continue to do this work. So there's also a training component. And there's also meant to be, with those government officials that came with us, a feedback component to the household. And so this, again, I think you raise a really good point, which is, again, so I went for like 2 weeks, right? But my 2 weeks was this intense time for me to help them do the swabbing and all that. But there are years of community engagement around my 2-week trip, right? And I wouldn't have gone or been able to go or ethically been able to go without that. And I do think there's an increasing push, and which I support, of all of these trips have a training component. So it's like, I did have to set it down. They wanted 15 government officials to go into each household with me. And I was like, I cannot take a group of 2 enumerators for the survey, 2 people to swab, myself, and 15 government officials. I can't bring 20 people into somebody's household. Like, I just can't. So I did cut it down to 4, which was still like a group of 8 people in the house, which I still felt was overwhelming. But it's a balance, right, of the ethics of all of this, right? And I think we need to think about that. I don't know our timing. How about, go fast, Julia?
I will take the hit here by asking, what's so bad about these mundane recommendations? You know, it's like wash and boring, what's complicated, but it's essential, basic, and has.
Those are emerging infectious disease people who work with like Ebola and Lassa, and a hand washing station is very boring. They want, yeah. Exactly. No, this is my point. It's the basic things that work. But when you're kind of thinking of, yeah, no. But we are fighting that. Like, the sense of it should be sexy. It should be novel. It should be innovative. That's like what the donors sometimes are asking for. Yeah. I don't know. Clarissa, was that your comment too?
I had, yes, one was what did they want?
Yeah, they wanted sexy, innovative.
I don't know what would be sexy.
I don't either.
That's why I thought I.
Yeah.
And then the other question I had was, you were looking for coronavirus, but were you expecting other pathogens as well?
Yeah.
I don't know what's in that one.
So this one, bats is coronavirus is the risk, but I've done a similar study in Liberia on rats, where we looked for Lassa, where we swabbed the rat tracings in the household and found rat poo was great. That was even funner than this. More PPE with Lassa, so it was hotter too. It was 108 degrees when I did the sampling. Full PPE. I think, okay, thank you so much. It's great happy to talk afterwards.
Please come find our presenters afterwards, and maybe we can do a sexy wash panel, Clarissa, next year at the conference. All right, and up next we have Anjerul Islam from UNC.
Hi everyone, thank you so much for joining my talk today. My name is Syed Anjerul Islam. I'm a second-year PhD student at University of North Carolina at Chapel Hill with the Aquaya Institute. So today I'm going to briefly talk about the sanitary inspection characteristics, precipitation, and microbial water quality, which is a 3-country study of rural boreholes in Sub-Saharan Africa. It's not forwarding. Oh, I can't see it here. Okay, I don't know, can I think there is something wrong with that. I am present.
These are your slides here, Zach.
Yeah, but it's not coming.
Oh, it's not moving.
No.
It's moving forward, is that, or what's wrong with it? Do you need the animation?
No, yeah, I need the animation for later slides here.
Oh, I see, okay. All right.
I'll take the last question because we have these 30
Yeah.
seconds.
Yes.
Perfect, thank you.
David was going to ask a fantastic question
like, oh, the bats never disturb us, never. The non-farmhouses are like, they boo, they do this, they do this. So it is a really interesting, I have to say, my assumption is totally wrong when I read this question.
Okay, there it goes.
The computer
Okay,
working?
cool.
We're good. All right.
Tony just pops out of every corner of it.
Sorry about the interruption. So the motivation and background of this study is that according to the recent GMP report, around 69% of the population lack safely managed drinking water services in Sub-Saharan Africa, which means that they don't have improved sources at their premise. And approximately 200 million people depend on boreholes in Sub-Saharan Africa region. And rural water safety in that region is a challenge because mostly it is managed by community, and then monitoring and managing water quality every day would be a challenge because of the transport and things like that. While sanitary inspection can be a valuable water source risk assessment tool. And climate change has increased the rainfall, it is going to impact the water source functionality as well. So if we can perform water quality testing continuously, then it's a very effective tool, but it comes with some challenges, which is it is really costly to collect the sample and then process it and then analyze it in the lab. Also, it will require much more training, and we are just collecting samples at one point of a time, so it might be variable depending from where we're going to collect the samples and what time during the day we'll collect the sample. Also, for rural water systems, because of the infrequency, it might be vulnerable over time. So we thought, our hypothesis is that the combination of sanitary inspection tool with the water quality testing would gonna better characterize, predict, and address water system vulnerability. So what is sanitary inspection? I know many of you already know that, but just to give a background, so sanitary inspection is a systematic evaluation checklist of risk factors for water sources. As you can see in the figure that the animals are roaming around close to the water source, and their feces may be pose risk to the water source. So it assesses water system vulnerability to fecal contamination. The couple of advantages of using sanitary inspection is a straightforward approach where moderate training required, and it's effective and low cost for risk assessment of the water sources, and also there are opportunities to refine and optimize the sanitary inspection questionnaire. So this is a great figure that I just want to quickly give you an idea what a sanitary inspection is, and you can see a source of like a hand pump connected with borehole is there, and then it is a fenced, there is a fencing around that, and also it connected with the drainage channel. And a sanitary inspection is a collection of questionnaire, and then based on yes/no responses, we can figure out the risk factors. So as you can see that if there is a latrine within 10 meters, then it's a risk factor of 1. And then question number 2 is if there is a latrine uphill, then it's a risk factor of 2. So also because I'm using different color and different shapes, just to let you know about different risk factors that we will subcategorize later. So number 4 is about if there is a stagnant water, and number 5 is there is a brokerage channel. And the triangle shape is about any fencing around the water source, and number 9 is the cement floor is cracked or not. And then we subcategorize these risk factors into three ways. Oh, I forgot about the rain. When the rain comes in, all risk factors are going to be much more worse. And so the subcategory is source, carrier, and the barrier risk, where the source is mostly if there is a source of contamination, uphill, latrine, or things like that. And then barrier risk is about any fencing around the source, or if there is a apron channel. And just a list of source, transport, and barrier risk factors. Source is a latrine within 10 meters, and then transport is if there is any stagnant water or drainage channel, and barrier is about cement floor and their conditions, apron and their conditions, proper fencing, things like that. I'm just going to briefly mention giving more time into that so that you have an idea about what source, transport, and barrier risk means according to that water sources. So based on that, we have developed some research questions. First one is if there is any association between sanitary inspections score with the fecal indicator bacteria if we consider rainfall into our model, and that for rural boreholes in Sub-Saharan Africa. If such relationships are observed, which specific risk categories are most significant? And can current sanitary inspection methods be optimized or refined based on the available data and evidence? So we collected data from Ethiopia, Ghana, and Burkina Faso, and mostly from the rural areas. And this is a cross-sectional study performed between 2014 and 2016, where 6 international NGOs, including One Vision, helped us to gather the data. Most of the training to the enumerators performed by the Water Institute professionals, and data is recorded on the mobile platform like EPCOFlow and mWater. So the bar chart does give you an idea about the observations that Burkina Faso and Ghana has more than 900 observations, where Ethiopia just have only 400. So the data is, sample is collected in a sterile 100 ml oil pack thou bags, and then it processed in the field using the compartment bag test. And we use compartment bag test because it can be used at ambient temperature. For the incubation can be done at ambient temperature, and depending on the temperature, the incubation time may vary. So for the quality control, we used random field blanks and duplicates. So E. coli concentration is listed as most probable number per 100 ml. And then we subcategorize these according to our WHO risk categories, where conformity means less than 1 mpn per 100 ml, low means 1 to 9, and then so on. So this stacked bar chart does give you an idea about our like MPN risk category, where we can see that mostly 50% of our samples within the range of conformity region where there is no risk, and around 30% or 1/3 of the sample is at high risk category, which is greater than 100 mpn per 100 ml. We also gathered meteorological data from which is globally available. For precipitation, we have collected data from Tufts dataset, and for water table depth data, we have collected it from a globally simulated data of Fan et al. Just to give you an idea, because we have used cumulative precipitation over last 48 hours from the sample collection, so for overall, we have got around 5 millimeter precipitation, and then by country, when it is disaggregated by country, we have got that Ethiopia has the highest rainfall. For mean outer table depth, we have got around 20 meter, and when we disaggregated by country, we have found that Ghana has the highest mean outer table depth, which means that Ghana may have extracted more groundwater, just an estimation. So we have performed both binary logistic and order logistic regressions to estimate the association between sanitary risk factors with microbial water quality. For binary logistic regression, we used E. coli as our outcome variable as presence and absence, and for order logistic regression, we used E. coli as the WHO risk category. For independent variables, we have used sanitary risk. We have aggregated, then disaggregated. Disaggregated means source, transport, and barrier, as we have mentioned before, and then individual risk factors. But like each question, how much that has an association with the microbial water quality, and then the meteorological data. So this is just give you an idea how we have calculated the risk factors. Based on the yes/no responses, we have just calculated the sum of all yes responses and then number of sanitary conditions applied. So here on the hypothetical example, we have around 7 yes responses and then number of 10 conditions applied, which means we have an overall risk score of 0.7. So when we run the model using the aggregated risk score, which means the overall sanitary risk score, we have calculated that, and then with the independent variable of cumulative precipitation and water table depth, we have found that for ordered logistic regression, it has an odds ratio of over 4, which means that if there is a overall sanitary risk score increases, then the probability of getting from lower risk to higher risk will be 4 times more likely. We also have found a significant association with cumulative rainfall, although the effect is not that much. So for ordered disaggregated risk score, we have source, transport, and barrier. We did not find any association between source and transport, but we have found significant association with an odds ratio of 3 and around 2.5 for binary and ordered logistic respectively. So, which means that if there is a barrier risk present, the probability of getting microbial contamination will be 3 times more likely. So this is the risk factor when we run the model individually for each questionnaire, and we have found for the both models, visible cracks on the cement floor and improper fencing has significantly associated with microbial contamination. And for ordered logistic regression, we have found drainage as channel and absence of cement floor and without extension has some significant association. And similarly for the binary, you have got 2. So just to give you an idea, we have run the we have about 20 questionnaires for that. We have run the model, but we only got around 7 questionnaires which have significantly associated with the microbial contamination. Couple of limitations for our study: we did not provide equal weights. We have provided equal weights to all questionnaires, but if there is a species close to the source, then it might have higher impact than if there is a latrine like maybe 10 meters from the ground. So we didn't we didn't figure out a way to give more weights to any certain questionnaire. Also, this is a cross-sectional study, and this is just a snapshot of a time. It might be helpful if we have a longitudinal study, and it can be more sustainable. And then the water table depth data is modeled, not directly measured, and that's also a limitation for a study. We didn't consider any topography into our model, so we have considered rainfall into our model, so that might be an impact on local topography. So the key takeaways for the sanitary survey, overall sanitary risks can predict water source vulnerability. Barrier failure is crucial and has the potential to predict water source contamination. Source and carrier risk may be difficult, but to reliably capture with the sanitary inspection, because sanitary inspection is just walk around the water source, so it might not capture all of that. So for precipitation, it can impact the water quality, but further opportunities to optimize the duration or lag over. So for our model, we have considered precipitation of last 48 hours, but it might be worth to look into like what's the cumulative, if there is any association between the cumulative precipitation of last 7 days or maybe 10 days, things like that. So we didn't find any association with the water table depth. And overall, we have found that ordered logistic model can be a better predictor than binary logistic model. And I just want to acknowledge the funding of our project from World Vision, WHO, and the Water Institute, but the primary data collection is done with the funding from Conrad and Hilton Foundation. We got some statistical support from the Water Institute, and I really acknowledge the project personnel and my advisors for this study. Thank you so much.
Time for a couple quick questions. I'll walk it around this time.
Thanks very much for that presentation. It was really interesting to see such a wealth of data on this. I think some of the things you might want to consider in your research are about how useful CHIRPS is for this kind of analysis. It's the reliability of it in terms of the 48-hour time period within the specific locations is probably not high. So you might want to look at what's more appropriate for that, and to consider whether, I wonder whether you actually looked at different time periods as well, because obviously there's many different pathways through which rainfall is going to be influencing water quality. But the other thing I wonder is, did you look at the potential for bias in the sanitary inspection results, or differences in sanitary inspection results by weather? Were they worse, or were there more indicators, for example, of water pooling as one of the indicators when it was raining, and is that perhaps one of the associations that's part of that data?
So, thank you so much for your questions. I think it's a great question. So your question is, if we have observed any biases on the sanitary inspection based on the rainfall data that we have gathered from the meteorological data afterwards. I think the sanitary inspection is normally done with using very moderate training. It doesn't require so much training. So, and the Water Institute professionals helped the enumerators to collect the data. But we didn't have any, I don't think if we have any biases in terms of from where we're going to collect the samples, from where we are going to take the sample, have the sanitary inspection that is helpful. So yeah, I didn't see any bias in that configuration, I guess. If I have you right on that. If I have the right questions.
Another follow-up to what Katrina's caution about chirps. Chirps is at 5 kilometer grid resolution, is
Yes.
that right?
Yes.
So yeah, my immediate sort of question is that the variability over the course of that 25 kilometer pixel could potentially, make the reliance on that variable challenging. But okay, this is a big jump. Tell me if this is an absurd kind of inference. But I think your coefficient for barrier damage seemed to be a dominant factor. So I'm trying to imagine what more evidence we'd need to collect to be able to simplify a sanitary inspection to just an inspection of whether there was barrier damage. Like if there's some indication that it's such a significant indicator or influence that we can just say, well, given limited time, given limited resources, that might be a reasonable jump. I realize that's it may be far-fetched, but I'd love to hear your thoughts on that.
Well, so previous study from Emma Kelly, she also got that barrier risk also made a significant impact on the predicting microbial water quality. So I think maybe I can just jump into, so when you look into individual risk factors, and here you can see most of them are actually the barrier risk, because for visible cracks and improper fencing and absence of a cement floor and without an extension of wall below the ground, and these are all barrier risk, and these have significantly associated with the microbial water contamination. So the objective of our study is to find how we can minimize the number of sanitary inspection questionnaire. And here we have run for 20 different questions, and we have found significant association only just 7. So I think it might be a start where you can just start looking for how other models can predict, or we can also go for other data if we have, then just run the model and see if they also have the significant association with this questionnaire, or if there are other shows up. Thank you so much.
Great. Thank you, everyone. Is this working? All right, up next we've got Kelly Baker from the African Population and Health Research Center.
Right. Actually, I'm at the University of Iowa, but like Danielle, I'm finding myself accidentally up front because the Kenyan government refused to give our project manager a visa to come to this conference. So happy to be here, but kind of sad that Phyllis, who developed this project, is not here to lead it. What I'm going to introduce is a protocol that Phyllis led, developing a geo-tracking method for understanding the sort of spatial and species interaction dynamics of how enteric pathogens might be transmitted. This is a part of a bigger NIH-funded study called Pathome, and I'll provide some information about Pathome at the end. So the point of Pathome is to really sort of explore in urban settings how children and animals acquire enteric pathogens and swap them back and forth between each other. We focus on children under 5 because they face the highest risk of contracting environmental and zoonotic pathogens in these environments. And the goal of the overall Pathome study is to predict, sort of where and how you might act in urban environments to prevent pathogen transmission via these different pathways. But the goal of this specific project was to develop this geo-tracking method that could be implemented in the broader parent study. Right, so some common approaches that are used for identifying human interactions with other humans, with animals, with the environment include surveys. It can include structured observation. Videography is a technique that's emerged in the last couple of years as well. But there are some major limitations to many of these methods, which include response bias to being observed or to being asked about sort of behaviors that you may already be aware of are not recommended. So combining public health research and geo-tracking methods can provide a lot of opportunity to reveal these sort of spatial temporal dynamics of infectious diseases and determine which processes or which sort of hygiene conditions in the environment are the most important for enteric pathogen spread and prevention. All right, so before I move on, I'm going to put the sort of acknowledgements up front here, because Pathome itself is this big project that spans many different disciplines and has many different people. I don't see a pointer, but this beautiful young lady up here in the far left in the white sweater is Phyllis, the one who led this project. And I also want to acknowledge that this is funded by the Fogarty Institute. Okay. So for this study, the goal was to develop a protocol for using geo-tracker devices to map infant and domestic animal movements. We focused on several different methodological procedures for using geo-tracking devices, which included sort of ethical considerations, the concerns of study participants, especially related to parents of infants about being geo-tracked, as well as community reactions. We also wanted to understand whether infants and animals would tolerate wearing these geo-tracking devices. And the overall goal was to develop some recommendations for adoption, not just for the Pathome study, but also for anybody who may be interested in a similar approach for studying other types of diseases. And in the end, we are sort of currently validating the protocol and sort of using this to refine our guide for other purposes. All right, so here's our stepwise methodological process. The first thing we did was to conduct a systematic evaluation of other studies in the literature that have used geo-tracking devices, as well as a market scoping review to look at what was available out there that would work in countries other than high-income countries, that would be affordable, that were small and lightweight in size. So we had a range of different criteria that we were looking for in these technologies. And then we also were looking for harnesses and other types of devices that we could use to affix them to infants, to animals, ranging from chickens to cats and dogs. We then took the sort of devices, so here on the left is the device that ultimately worked best in our study. It's made by a company called Tractive. We took these devices into our study neighborhoods and held community engagement meetings to talk about our intended use of them and to gather information that we could use to revise our protocol before we ever even went in for pilot studies in households. We conducted a 24-hour pilot study next, using that community-based sort of protocol to track infants and the domestic animals that they lived with. Here we were looking at factors like whether or not placement was challenging, tolerance, right? Were animals and infants sort of agnostic to wearing these devices? Could we recover the devices after we had placed them, or did they get lost? Let's see, what else did we look at? Battery time, how long could we leave these on? And we adjusted through this pilot process certain things, like, for example, how we fit devices onto animals or children. Then we evaluated the technical and data management aspects of protocol design, sort of really looking at consistency or reliability if we wanted to scale these up as a technology for our broader cohort of households and infants. And then finally, we conducted exit interviews among caregivers as well as community health volunteers, because these were people that agreed to participate in the study, and they obviously, through that experience, probably formed unique sort of insights into what it felt like to participate in that study, and we wanted to document that. Oops, let's go in the opposite way. All right. So this is just an example of some of the feedback that we got from the stakeholder engagement meetings. Some of the sort of items that we heard about are things that we did not react to in terms of changing our protocol design because we could explain them. So for example, this top one was concerned by the community about whether trackers were waterproof or heat-proof, and so we simply sort of repeated information about the durability of the technology design. There were other comments that we did modify our protocol in response to. For example, number 2 is difficulty in caregivers choosing an appropriate and comfortable place for putting trackers on infants or animals. And what we did is we created some flexibility in our design where we let caregivers select where they wanted to place them, particularly on infants. We ultimately did not need to change it for animals. Another one to point out here was whether the device posed any health risk to the infant or the animals being tracked. We did not stop the study, but that was because we sort of responded with health information that's provided by this commercial geo-tracker. We modeled the wearing of these devices ourselves to demonstrate safety and explained that it's basically nothing other than a smartphone. I mean, that's all it is a case with a GPS tracker and a telecom chip. All right. So from the pilot study, when we were looking at acceptability, what we found was that caregivers generally embraced the geo-tracking exercise at first solicitation of interest and participation. A few of them did express some concerns about consequences and beliefs about geo-tracking over the course of the study. Most of these cases were resolved by sort of discussion with community health volunteers. The device's physical form also influenced acceptability, and so we worked out some ways to make them unobtrusive, including sort of bland colors for harnesses, and again, caregivers could choose where they wanted to put them on the infant. We did not have any concerns from household owners about placing trackers on animals, other than the concern about their neighbors seeing their animals walking around with geo-trackers. And so, we made an effort through CHVs to sort of dismantle, we informed the community about what was going on and why we were doing that. So we did sensitization, but then also, we tried to sort of bland down what the device looked like on these animals. All right, in terms of approach and compliance, with preferences on where the tracker should be placed. For infants, caregivers preferred inside the pockets of infant clothes or around the waist or wrist or upper arm. It's really kind of all over the place, between the child's pants and diapers. With animals, we placed it around the thoracic or abdominal area, although that's pretty regulated by by animal subjects review boards. The infants' reaction to the trackers was no sign of discomfort. A few of them were initially curious, but otherwise they largely ignored these devices. You can see a picture up in the left-hand corner of the tracker that is near a child, that was participating in the study. We did give caregivers the options of putting these trackers near the child instead of on the child, if that was what they wanted to do. With animal reactions, some animals showed discomfort in the beginning, but by 30 minutes later, they had also adjusted to the devices. The devices were efficient and effective. We didn't have any issues of battery depletion. They all lasted for the 24-hour period of geo-tracking. There were some delays in devices detecting the GPS signal when we started them up and placed them in indoor settings, and we fixed that by having people go outside to initially place the tracking device. And we did collect sort of data, reliable information about movement of the devices, and we're able to monitor it in real time on the Tractive website. This is an example of some of the maps that you can generate by looking at at the device is in real time or retrospectively on the child. The one that's third over is a 24-hour time-lapse map, so it's showing all of the locations that the subject wearing that tracker went in that period of time. From the sort of exit interviews, we found that the initial concerns about privacy or sort of change of behaviors, that we did hear some comments about caregivers being concerned, specifically around the devices showing red or green lights and whether it was recording them visually. So they were worried about it more from a videography standpoint. And so one of the things that we've done is try to mask the lights, and then also we reassured them that there's no camera, going back to the message about it being like a smartphone. With animal distress, some of the caregivers did report noting animal distress during periods where we were not observing the animals. But when we went back to the households to recover the devices, we did not observe these types of issues. Key technical challenges were devices being accidentally switched on or off by enumerators, occasionally signal issues, especially in indoor settings, fitting harnesses and resolving sort of animal distress around wearing these devices. And there were one or two times where we sort of lost trackers. I'm going to save that for questions for the sake of time. So we have a manuscript coming that's going into peer review that's describing some of our key recommendations from this. I'm not going to sort of go through them in itemized fashion, but generally we concluded that we were able to work with community health workers and leaders to enhance acceptability, and that included modeling wearing the device. We conducted much more rigorous training in the parent study of enumerators on how to use the devices properly. And so we do believe that these satellite-based pet geo-tracker devices can be used for human-animal environment research to reveal transmission pathways. There's a lot of other types of One Health approaches that this could be useful for studying spatial and behavioral dynamics. And we will, over the course of the parent study, generate a lot more data about the validity of this information and the advantages and disadvantages. This is my final slide. If you're interested in the sort of broader pathome study, we do have a preprint online. It's coming out in press in BMJ Open this month. We have a somewhat underdeveloped website, but more immediately, we have a side session tomorrow where we are going to be doing much more hands-on workshop-based activities, letting you explore these for yourself. And then in the afternoon, we are going to have outdoor drop-in and play activities, so just look for the chicken.
Was there a great talk, by the way? Thank you, and go Iowa. Love to see the representation, Dr. Baker. But I'm curious to know if there was any concern about like behavior change amongst the caretakers, like knowing that this study was about infant and pet and animal interaction. If there was any concern that like they might start to think more about that interaction and therefore segregate their infants from animals just purely by being involved in the study.
Absolutely. We did not find any studies like this specifically prior in the literature. So, we're kind of flying blind on this. But one of the things that we're doing to address that question is when we place the geo-trackers over that 24-hour period, we also have 2 5-hour periods of structured observation. So we'll be able to take that chunk of time where the observation and geo-tracking is happening simultaneously and compare what we observe in terms of interactions and going from outdoor to indoor areas. We'll be able to compare that to periods where we don't have observers in the home. It is possible, though, that this functions just like other methods where it does influence behavior. And so we do intend to explore that.
Hi, this is Nuhu from ICDDRB. So the objective is to reduce the bias of observation by the enumerator or video surveillance or other things. But it seems like from your interview and from your results, it added a lot of behavior changes and communication, particularly the community sensitization and other issues. So that's one of the issue. I'm not sure how this will be overcome while the next study. And how you come with the design of this geo-tracker. So is this the final product, or who actually designed this? Because for designing any device, you should have some process and come up with a final work. And you mentioned repeatedly about the smart watch type of things, but this doesn't look like a smartwatch. Why you didn't prepare this as a smartwatch, or like kids usually wear like ring or anything else? So that could be another question that can reduce the behavioral aspect within the household. Thank you.
Yeah, thank you for the question. To clarify, so this is a commercially available product. We did not design it, and that was purposeful. I'm not an engineer. And so what we were looking for were products that were on the market, available in most of the countries around the world, that anyone else could also go grab and do for themselves. And so the device design, it's actually kind of a design that's similar across a lot of different pet tracking companies. But it's, we'll have them on display tomorrow, but it's pretty small and lightweight. So we just didn't see the need to try to re-engineer something and go through the whole scale-up issue. A smartwatch would be another way of doing this. I think smartwatches are more expensive. So these devices are about $40 for the actual device. And then for a long-term year contract, we pay $6 to $7 per child to extract all of that information. So it's, a long-term for a year's worth of information. So there's just certain things that we couldn't do, and that's why we went with commercial materials.
Interesting, yeah, thank you.
Other questions?
Great presentation. I've seen a lot of work in this space in infectious disease, and I'm curious if you have intentions to partner with other people doing this same work, but for zoonotic vector-borne diseases.
Right. So, we are. This is a part of a much bigger infectious disease study. But as far as we did look at a lot of the geo-tracking technologies that are used more in the wildlife space. Following sharks and birds and things like that. And each of those had different types of challenges with adoption. So, for example, some required a pretty large receiver in order to detect the device coming close or far away. There are some devices out there on the market that provide even better GPS data quality, but they're $2,000 to $3,000 each. So we went through and we started with a lot of the ecological literature first to see if those would solve our needs, and we just wound up settling on the commercial devices because of their accessibility and cost and utility. Did I answer your question?
Yeah, like layering this with heat mapping is something that other people are doing in spaces, so I'm just curious if that could be a space as well.
Yes, absolutely. And is there 1 minute? Or okay. I can show you one thing. Phyllis really wanted to show this. So this is one of the byproducts of what we generate with this, and it can be made as a heat map too. So this is a video from two different 24-hour time-lapse data collection exercises with an infant in blue and the chicken that lives with that infant. And so we're able to overlay these pieces of information and look at how often infant leaves the home and goes out into the street. We can look at how often they're co-locating within a certain spatial range of accuracy in the household setting. But this can be heat map based data as well. Yes.
Great. Thank you, Kelly.
Thank you.
All right, and last but not least, we have Anna Marie with Aquaya. Take it away.
Thank you. So pleased to be here today to present this work on wealthy households drinking safer water. And I'll say, from the beginning, there were a lot of people involved in this work. So Aquaya, as well as Stanford University, PMA in Ethiopia, as well as Bahir Dar Institute of Technology, as well as KNUST in Ghana, and EGREC, which is in Uganda. So some background here, as of 2022 globally, we know that over 2 billion people lack safely managed drinking water, and that includes over 400 million using unimproved drinking water sources. And there's a lot of data out there on inequalities of drinking water access. So this has been well documented in terms of richer and poorer countries, Also urban and rural areas within those countries, and even to the household level of higher and lower socioeconomic status. There's been less research on drinking water quality and inequalities related to the water quality. I will mention that there is a fairly large study that was done a few years ago that looked at across 27 countries. So I'm not saying that there's no data out there. There is, again, that big study, but that was looking at it was nationally representative data, right? So we're looking at entire countries in 27 countries. And so what we're looking at for this, this project is to quantify wealth-related inequalities in microbial drinking water quality more at a local level. So looking at within individual communities or smaller areas. And this work was done in Ethiopia, Ghana, and Uganda. All right, so into the methods. So we did surveys at over 1,400 households, and this was across 6 districts within those 3 countries I mentioned before. And so the sampling was done with this 2-stage process. So we sampled 24 enumeration areas within each district. Then we did a household listing exercise, so basically a census of all the households in those enumeration areas, using the demographic health survey methods. And so did a standardized wealth index calculation for each household. And then we assigned quintiles, so 1 through 5 quintiles at, again, the enumeration area level. So this is like a community or a section of a city, something fairly small. And we sampled 2 households per quintile for the survey. So the surveys included observations of household storage, questions about water handling, and we asked households to provide a cup of drinking water, which we then sampled and tested for E. coli using membrane filtration methods. All right, in terms of the analysis, so again, we're looking primarily at inequalities and 2 main outcomes. So I'll present outcomes of water access based on improved and unimproved sources, and then the E. coli results. And we did this in like 2 main ways. So the first thing is a concentration index. If you're not familiar with that, it's a measure of inequality of one variable versus another variable, and this is used a lot to look at inequalities in health outcomes. And then the second analysis we did was a mixed effects logistic regression. And so this is looking at an outcome of low or moderate risk, so up to 10 CFU per 100 mil of the drinking water sample by wealth quintile. And this was controlling for a number of variables as well. All right, so into the results. So here we have just a few summary statistics. I won't read them all to you, but just to say again, it was about 1,400 households for surveys, a few less with E. coli samples. I'll point out a couple things. We have a high percentage of rural households, so it's not entirely rural, but mostly rural. Looking at the water storage practices, we saw some variety here between the different countries. So fairly high safe storage was seen in Ethiopia. That would be like a jerry can with a small opening and a lid. And then much lower in, let's say, Ghana, where people are more likely to use a large container with a wide opening or maybe not covered. Household water treatment, this is reported treatment, was quite low overall. The highest was in Uganda, but only 20%, most of these were boiling. In terms of water sources that were sampled, we had a wide variety. And so you're seeing here the 3 different countries. Hand pumps were well represented by all 3 countries. But we saw that in Ghana and Uganda, we had more piped water, and then in Ethiopia, more unprotected springs. So looking at this now kind of aggregated, but just improved and unimproved drinking water sources across the countries. So we see use of improved drinking water was lowest among the Ethiopian households by a fair amount, and fairly high in Ghana and Uganda. So for the E. coli results, these are categorized in the graph by different risk level categories. So where the darker color is like the higher risk, and again by country. So we saw overall 90% of household samples had positive E. coli results, and about half were in the very high risk, so over 100 CFU per 100 milliliters. Looking at individual countries, with those bottom 2 categories, so the safest 2 categories, it was about 10% from Ethiopia, just over a third from Ghana, and about almost a quarter from Uganda. So we also saw kind of worst water quality in Ethiopia. All right, so now I'll kind of go back through those results by the wealth quintiles. So we see for the types of water sources here, improved and unimproved, that the lower, the poorer households, the lower SES status, so that's the ones on the left, like numbers 1 and 2, were more likely to use the unimproved drinking water sources as opposed to the households in the groups on the right, the wealthier households. So this is a concentration curve that you'll see on the right here. So how to read this graph? If that curve were right on that dotted line, so at that diagonal line, that would mean that there's kind of no inequality, that everything is equally represented. What we see here is that it's slightly offset. And if you take the area between that curve and that line, you get what's called the concentration index. So in this case, it's a negative value because it's like on the left side of that graph. And that's telling us that there is disproportionate distribution of this outcome of using an unimproved source between the different wealth quintiles. And we see this, in this case, it's statistically significant because our confidence intervals are all negative here. So now looking at this by country, what we actually see is that Ethiopia is driving those results. So the last graph was everything put together, and now we're separating it out. So if you look at the graph on the left is Ethiopia, and we can see visually a much bigger difference between those wealth quintiles, as opposed to Ghana and Uganda, which are fairly consistent across the different wealth levels. The concentration indices are listed at the bottom. So Ethiopia has the biggest number, the biggest negative number, which means that's, you know, the most inequality, and that one's significant, whereas the other two are not significant. Okay, so shifting to the E. coli results. So we're seeing something quite similar when we put them all together. So the households with a lower economic status, so again on the left of this graph, had higher levels of microbial contamination. And similar here with the concentration curve, again we see a negative value of the concentration index, which is further confirming higher levels of microbial contamination are present with poorer households, lower socioeconomic status. Okay, so now this one is a little bit different from the last one I showed you. So before we saw that the improved sources were varying only in Ethiopia and not the other two countries. But here we're seeing differences in all three kind of subsets of our data set. So across wealth quintiles, just sort of visually squinting and looking at this, we see that they are moving down from left to right. We also see that confirmed in the concentration index, which is shown at the bottom. So in this case, actually, Ghana had the highest variability between those wealth levels, followed by Ethiopia, then Uganda. And then looking at the regression model that we ran. So again, we're seeing in this case, the wealthiest quintile, so the number 5, had better water quality than the poorest quintile, number 1. So we didn't see a difference in the ones in between. So like 2, 3, 4 were not any different from the poorest quintile, but we did see a difference between the wealthiest and the poorest quintile. And this is controlling for a whole host of things, including the source type that people were collecting from, their storage and treatment practices, urban and rural setting, population density, precipitation, which we heard about earlier, and even clustering at the EA level. So we also did a kind of a subset regression to look at so let me give an example of one thing we saw within Ghana in particular. There were a lot of households using sachet water. And when we looked at who was using the sachet water, maybe it's unsurprising, it was the more wealthy households. Also, the quality of that sachet water was higher than most of the other sources we had. So there could be some interactions going on here. So as sort of like a sensitivity analysis a little bit, we pulled out some of our samples. So we pulled out all the sachet water, and we looked at just a subset of improved sources that looked like they were fairly consistently used across the 5 quintiles and also had fairly consistent E. coli results. And so our sample size dropped a fair amount down to about 500 households. So it's a much smaller sample in this case. But we're still seeing some differences. So the wealthiest quintile was more likely to have safer water than the poorest. Although I will say in this point, this is only true for the higher risk levels, so over 100 CFU. All right, so just to summarize here, we did see, widespread microbial contamination in our household drinking water samples in the settings where we were working, particularly in Ethiopia. And water contamination, disproportionately affected the poor. And this is what we're seeing is, for reasons other than just the source types that they're using, or maybe the water handling practices they're using. And I do want to point out also something I said earlier about the sample, right? We're seeing this from even the wealthier and poorer households living in the same communities. So we're not talking about the richest household in the city versus the poorest in the rural areas. I think that to me is one of the main findings that's a little bit different here, is we're looking at small differences perhaps in the wealth, but still carrying through to water quality. And so yeah, there's a need for future research to really understand what's underlying these inequities, and as we strive to leave no one behind, that there's perhaps reasons or areas for targeting interventions to these poorer households. So I'd like to thank the Hilton Foundation who funded this work, as well as all the partners I mentioned before, the data collection teams, and the survey respondents who kindly gave us their time. I'm happy to take questions.
Yeah, an excellent presentation. I have two questions or suggestions as well. So, have you analyzed your data using the type of sources, like which source are contaminated more? I didn't see that in your presentation. Number one. Number two is if further analysis on the disease of this community, like Ethiopia, Ghana, and Uganda, that what are the diarrheal prevalence of these areas, whether there is any correlation with the diarrhea and water contamination. That would add the value, and you can also report the government that this could be one of the association. Thank you.
Okay, thanks. We did not look at disease data, so that would be interesting to overlay with this. So I don't have that on the top of my head. In terms of the different water sources, I did spare you previously from the full, very small, full regression results, but I'll put it up here now because you asked. So we did see on the left are the water source, water point types that the samples were collected from. And so we did see actually unprotected springs were lower quality, and bottled or sachet water was higher quality. And then the rest, and the comparison group is a borehole with hand pump. So the rest were not significantly different. But again, this is not sampling from the water points, it's at the household.
Fascinating presentation. Thank you very much. I was wondering, what was the use of looking at improved and unimproved water sources? And do you also have data on E. coli contamination of those two sources? Because I would be very much interested if there's a difference in E. coli contamination between improved and unimproved, because I didn't see that coming back. And my other question would be, could it be that the more wealthier households would have more running water in their house, or maybe more access to soap or a toilet, so that there are wealthier households have more hygiene practices, which might now influence the water quality? Did you look into that, or? That was my two thoughts I had.
Okay, so I'll start with the second one. Our surveys were fairly targeted. We didn't have a whole lot of additional wash indicators, but I'll have to go back to see, to try to pull out some of those other differences that may have been between the wealthier households and the poorer households. Because I think there are a lot of things like that, like hygiene practices, that may also vary with wealth. So that's a good point, thank you. And then the first one was about the quality between the improved and unimproved sources. So I think they're not grouped in that way in this table, but it's reflected a bit. I guess the two examples that I gave before would be the unprotected spring as an unimproved source versus, the borehole with hand pump, which is an improved source. We did collect samples at water points as well, which I don't have here as part of this presentation, but there were differences in the water quality at the water point as well between the improved and unimproved. But that's not to say that all of the improved water points were safe to drink. Yeah. So there was still high contamination within the improved ones as well. But they were different, but yeah.
That's still very interesting.
Anna, that was really interesting and a really vigorous analysis, but I wanted to come back to some of the earlier points about making research sexy for donors and challenge you on this idea that it's the wealthy have better water quality. I think your results show that no one had great water quality in there. And perhaps, is this then a useful analysis if you're trying to pretend there's a group in this society who has better water quality when in fact, uniformly, I think you're saying there's 15% maybe who have good access. It's yeah.
That's a great point. And yeah, this is not good for anyone, right? We have, this one. Yeah, only 10% of our whole population had household water with no E. coli. And so, so it's a challenge. We're still remaining with the challenge of household water quality that's low across the board for everyone. So yeah, I don't have any brilliant response, but that's a good point. Yeah.
My name is Peter Jensen, University of Copenhagen. Thank you for a very interesting presentation. And we have seen similar stuff in a big study we did together with Nuhu in Dhaka. But what we see a lot of this is where it comes to water quantity. And this is sort of what I lack a little bit in this one, because, I mean, even though we have the different sources, improved, unimproved, it doesn't actually say how much water you have access to in your household. And we have seen that access totally determines the level of contamination that we see in the point of use. I mean, in the drinking water glass, nearly despite whatever level you have, so water quantity is equal hygiene in the household.
Mm-hmm. Yeah, we did not collect water quantity data as part of this, or at least it's not part of this analysis. I think we may have it, actually. Yeah.
All right.
Thanks.
I see lots more questions, but I'm keeping everyone from the happy hour. I'd love, if you have another question, please come grab our presenters. They were willing to hang out for a few minutes. But thank you all for the great questions, and let's give our presenters one more hand.